Yep, so my name is Ying, and today I'll be presenting my talk titled Rediscovering the Leaf Blotch Minor Moths of Australia. So they are in the Lepidopteran family of Gracilariidae. So I'm a PhD student based at the Australian National Insect Collection, which is in Canberra on Ngunnawal country. So I'd just like to take a moment to acknowledge the traditional owners of the land of which I'm presenting from. So I pay my respects to the elders past, present, and emerging, and I extend that respect to any Indigenous people watching today. So just briefly, I wanted to go over what is a leaf miner. So in general, a leaf miner is an insect whose larvae feeds internally in leaves. It doesn't describe a taxa, it describes a life history. So the leaf mining behavior occurs in the larvae of 4 insect orders. So it's quite a diverse, so it happens in quite a diverse behavior. So it occurs in Hymenopterans, Coleopterans, Dipterans, and Lepidoptera. And broadly, the diversity of species goes in that order as well. So the Hymenopterans are the least diverse and the Lepidopterans are the most diverse. So because of all this, because so many different insect groups leaf mine, these leaf mines can be quite distinctive, diagnostic, and also reflect their life history. So just as an example, you might have sap feeding larvae feeding at quite, or you can have larvae feeding at different levels just within the leaf tissue. So you might have sap feeding larvae who feed quite high in the tissue layer, and they produce these silvery mines that can only be visible on one side of the leaf. So if you flip it over, you won't see any damage. Alternatively, there are also these tissue feeding larvae who feed a bit more indiscriminately within the leaf, and when you hold it up to the light, you'll actually be able to see through and see that larvae inside. So just I wanted to share some interesting images of different types of leaf mines that you can see. But when you look at the last 5 images, they're just from Gracilariids. So even within one moth family, you can see that there's a lot of diversity in how the leaf mines appear. So what about my taxa? So I'm studying the Gracilariidae. They're also known as the leaf blotch minor moths. So they're the most taxonomically and ecologically diverse of the leaf miners. So there's about 2,000 species globally, but within Australia, we estimate that, there's about 200 described, but there's many more undescribed species, and that number will definitely change as my PhD goes on. And these moths are really quite beautiful. So when you look at them at a high magnification, you can see that they have these beautiful patterns on their wings and that they hold them up against their body, and they kind of have this interesting upright posture. So within this group, there are some species that are used as biological control. So for instance, against Patterson's curse, or they were introduced as biological control. And some species have kind of like a pesky behavior. So if you have citrus plants, you might be keeping an eye out for citrus leaf miner. So this is the kind of leaf mine that citrus leaf miner makes. It's a bit of like a winding type of mine. And unsurprisingly, therefore, the common name of this group does not reflect the behavior of all the species. Like this species doesn't make blotch mines. So another interesting thing about this group is that the larvae are hypermetamorphic. So the larvae have 2 distinct forms. So they do go through a number of different instars, but the instars can be divided into 2 forms. So they have this flat form, which kind of feeds at that sap feeding behavior, and they're really flat. So there's this example of this larvae, and that's on a piece of paper, and it's not actually like all dried up. That's actually how thin it is. And then as it develops, it may turn into this round tissue feeding form that is kind of more cylindrical and indiscriminate about what tissue they're feeding on. So as you can imagine, something that lives inside a leaf is really small as an adult. And that means that they're quite hard to observe. So that's it right there. That's the adult. So something fun that I thought I should cover was just looking back at some of the taxonomic research of on Gracilariids in Australia. So when we all think about the different groups we work on, it's kind of, there's not really, highly unlikely that there's this huge lineage of who has worked on it. So the first monograph was written by by Turner. So he did that in 1940s. And that's actually the only Australian monograph on Gracilariids. And then more recently, Ian Common, who literally wrote the book on the Australian moths, he did some collection and also read some Gracilariids. But the big gun in terms of Gracilariid research in Australia was actually Dr. Toshio Kamada. And he read a lot of specimens and did a lot of collection and dissections, and we have a beautiful collection at ANIC thanks to him. So what's the current situation in terms of the grassariid phylogeny? So globally, there are currently, as of a few months ago, there are 9 recognized subfamilies, but the Australian fauna is still divided into these, or organized into these 3 subfamilies. So the Gracilariinae, the Litocoletinae, and the Phylloxanistinae. And so when we think about this, this paper, a lot of the data that was in that paper was actually global specimen samples and did not include the Australian stuff. So because of that, we don't actually know where some of the endemic Australian species and genera might actually fit within this whole phylogeny. So that's something that I'll go into about my own project in a moment. So in addition to that, when we think about the state of this group in Australia, there's actually no tools to identify these micromoths. So as I said, some of them are pests and some can exhibit pesty behavior, and some of them feed on a lot of different plants, but there's no ability to really identify them with either molecular or morphological tools. So that Turner paper in the 1940s, it had no illustrations, not even of genitalia or wing patterns. It was literally just descriptions. So it's a very difficult thing to refer to. So that kind of is the crux of this project. Like, how can we identify a grassariid if you come across it? So overall, my project will increase our understanding of what of these moths. So the first or the biggest aspect will be restructuring the Australian grassariid. So trying to figure out where, within that current classification of 3 subfamilies, trying to fit all the genera into the now 9. And so that will be done using transcriptome data. And then I'll focus on 2 subfamilies. So I'll be focusing on the Litocoletinae and the Phylloxanistinae, and looking and trying to revise those 2 subfamilies to kind of provide or produce some more detailed morphological tools so that people can have a better chance of identifying them. And finally, I'll be looking at the host plant usage and the life history of this group. So there's a lot of literature out there with bits and bobs just like spread out about what they were reared out of. And Kumara himself did a lot of rearing, and I'm doing a lot of rearing. So I think I'll compile all of that, and that will support the other 2 sections of this project. So for instance, for the transcriptome data, I'll need some live specimens. So rearing them out would help, have this whole picture about this group. So to reiterate, that phylogeny has 9 current subfamilies. And so I'll be focusing on the Litocoletinae and the Phylloxanistinae. And so overall, I'll be using the ANIC collection to kind of take tissue from them and use them to create a new tree. So in terms of the subfamilies I'll be targeting, these were really chosen because they're a little bit more manageable. So there's only a few genera in the 2 subfamilies, but that's likely to change. There's a lot of unidentified stuff. So I think that there'll be a few more in the end, but I just wanted to highlight that these things are really, really small. So that's my fingertip, literally took that photo today. And then there's a specimen that you might be more familiar with, the Helicoverpa amicura up the top. And my specimens are really very small, but under the microscope, they look really, really pretty. So I just wanted to use this as a case study for how difficult it is to actually identify them. So this paper was written by Kamada and Marianne Horak here at ANIC. And what they did is that they looked at these 2 species of Dialectica. So the bottom species was introduced to as a biological control for Patterson's curse. But then they realized that maybe there's something already here that's kind of looking very similar. So they wanted to investigate this. So looking at the morphology externally, they looked at the wing patterns and they found that there were very little differences, and that wasn't very useful. So the next step for lepidopterists is that they look at the male genitalia. So as George Bin said in her presentation, when in doubt, get the Aedegus out, except here it does not apply. The male genitalia were basically, indistinguishable. So the next step, maybe look at the female genitalia, except here, again, it was also, they said in the paper that it was distinguishable, but to me, it's very difficult. And I don't think it's the best feature to use. However, when they looked at it in the life history, they found that the cocoons were actually quite different. So when you look at the upper picture, what you see is that the cocoon is made out of 2 layers, so the upper layer and the lower layer, and the lower layer is a bit more round and also white in color, and you can kind of see it's a bit more opaque. However, when you look at the lower cocoon, the upper layer is much more transparent, and then the bottom layer, the cocoon is brownish in color and kind of tapers off at one end. So that was consistent within the moss species across different plant species. So finally, I just wanted to reiterate that an integrated approach is required to revise this group. So as you can see, we need molecular data, morphological data, and life history information. And together that will help, provide some good tools to identify this group. So what I've also done is some rearing. So I wanted to show some fun little videos that I've recorded. So as I was saying, this group, they have the hypermetamorphic larvae. So this is the early instar, which are sap feeding. And they have these really cute little jaws, and they just chomp away at the upper layer. And you can even see that the rest of the leaf underneath the larvae. So they're really just feeding at that quite high tissue layer relatively. So then as they develop, they may turn into this rounder, larger tissue feeding form where they're just chomping away at the leaf, and they're just leaving the cuticle layers. So that's when you can actually start seeing them with the naked eye against the light. So yeah, that's just some fun videos that I've acquired. And so what are my future plans? Well, broadly, it's really to start doing some molecular work and build some trees from that. And I will continue rearing my leaf miners and going around looking for grassariid specimens in museums and collections. And I also just wanted to kind of advertise my iNaturalist project. So just this citizen science platform where I'm encouraging people to, if they do seek leaf miners, to just upload an image, and that will kind of add to the overall understanding of what leaf mines look like on all plants. And then if I find any that I find are very grassariid in appearance, I have reached out to some people and they have actually sent me some leaf mines. So I think it's a really good way to engage with other people, other scientists, other naturalists, and it's been quite successful. So if you are interested in collecting leaf mines, I would be happy to chat to you. So finally, I just want to thank and acknowledge everybody, my supervisor, Andreas, and people at ANU and the ABRS for funding my project. And if you would like to get in contact, please email me, tweet at me, Instagram me. I am available on everything. Thank you.
Thank you very much, Ying. Very nice talk.
Thank you.
As a boring natural entomologist, really nice to see that behavior and the behavior is important, not just more for phylogenetics. So
we need more papers like that.
Oh, we live in an era where people seem to have forgotten some of these things. But anyway, I don't think we really have time for a question, so please contact me.
Yeah, you can post in the chat and I'll respond.
Yeah, sounds good. It's actually now my